Today we will start with the starch. Starch, as you know that it is made up of what? Two component, two distinct polysaccharide are found. One is amylose, another is amylopectin. So starch is synthesized by certain enzyme and that enzyme called as starch synthase in the chloroplast. This is for temporary storage. Whereas in case of another component that is amyloplast in seeds, roots, and underground stems for longer term storage. So in examination, you will find two questions. One may be for starch as a temporary storage. Give the example. Starch as a long-term storage. Give the parts of plant or varieties. So first question's answer is starch is synthesized by starch synthase in the chloroplast for temporary storage in the amyloplast in seeds, roots, and underground stems for long-term storage. Now the starch granules. Starch granule contain two distinct polysaccharide types. One is amylose, another is amylopectin. So you can see here also how it is synthesized in the cytosols, in the chloroplast, how it is hydrolyzed. So amylose, as we know that, is a linear polymer of glucose residue that is linked by alpha 1,4 linkage. So n number of glucose is joined together in the polymer form. So residues with the alpha 1,6, wherever you will find alpha 1,6 branch, that point every 20 to 30 glucose residues on an average. So regulation distribution of the branch point within amylopectin, now amylopectin, that allowed the formation of organized array that is close to the packed left-handed double helix in the semi-crystalline zones of the starch granules. So essentially linear amylose is probably present so-called amorphous matrix of the granule and that also contain amylopectin. As I said, beta 1,6 linkage is less ordered structures. So the variation in the starch structure, you can see with it any biological source or any wherever you will find starch, there are a variation in amylose and amylopectin proportions. So another things, what are the differences is found in any starch component? There are three things. As I said, amylose and amylopectin. Another, the parameters or you can say parameters as a chain length. How much chain length distribution found in that? How much the degree of branching is found? Where does beta 1,6 at what component? 26 to 24 or 24? And another things, if you see under the scanning electron microscope, what are the granule size? This is all about the star synthesis. Now we are coming to these pictures. Here you will find chloroplast, another is cytosol. Now how yellow one, I am starting with the starch. But what is the title? Starch synthesis. So that much, as I said previously, glucose molecule is to be formed or utilized. So enzyme responsible for that is starch synthase. So glucose 1-phosphate, that comes from where? As I said, in glycolysis also, and that glucose 1-phosphate to glucose 6-phosphate, these steps comes under. So hexose phosphate isomerase, fructose 6-phosphate, fructose 1,6 biphosphate, these are the steps you will find in glycolysis. Now triose phosphate. How the triose phosphate is formed? Arrow it can indicate Kelvin cycle is operating. So what are the component you will find in the chloroplast and how it is translocated into a cytosol? Which component is outer? So triose phosphate through the enzyme that is called aldolase. Okay, wherever you will find 6,2, 5,1, 5,2, 5,4, 5,7, that wise the stepwise procedure is happening. Okay, so aldolase is the enzyme that will convert into a fructose 1,6 biphosphate. Now enzyme is biphosphatase, then fructose 6-phosphate that will convert into a glucose 6-phosphate. Phosphoglucoisomerase is the or mutase is the enzyme and that will convert into a glucose 1-phosphate. Here is glucose 1-phosphate, here is also glucose 1-phosphate. But here we are producing, this is found in the starch, chloroplast. As I said, in chloroplast for temporary storage. This is for temporary storage. Now, in the amyloplast or in the seeds, in the roots and underground stems, now triose phosphate come into a glucose, UDP glucose pyrophosphate and UDP glucose plus glucose that will convert glucose plus fructose that will convert into a sucrose. So sucrose synthase or sucrose phosphate synthase found in the cytosol. So which are the enzymes found in the cytosol? So you have to write sucrose phosphatase or sucrose phosphate phosphate synthase, and you can also write pyrophosphorylase or phosphoglucomure. These are the enzymes you can write in the cytosols. Okay, so this is all about the starch synthesis. Now, I have already told you how the fructose is formed in the cytosol. So we will read this, how this synthesis of fructose start. Most of the fructose found in the plant originated in the carbon that assimilated during the photosynthesis. So in photosynthesis, you know very well, carbon dioxide is fixed in the chloroplast via Kelvin cycle and it will form the triose phosphate. See here, triose phosphate. Now triose phosphate may be exported to cytosol. Okay, here this is the Kelvin cycle where it is operated, found in the plant, where during photosynthesis. Photosynthesis happening in the chloroplast. Now this triose phosphate transported to cytosol and triose phosphate may be exported to the cytosol where two triose phosphate molecules, how many? Two triose phosphate molecules are combined to create a one molecules of fructose 1,6 biphosphate. And these fructose 1,6 biphosphate that can be now the reaction dephosphorylated to form fructose 6 phosphate, which is isomer to glucose 6 phosphate, and glucose 6 phosphate can be used to form nucleotide sugar, that is UDP glucose. Here you can see, so UDP glucose and fructose 6 phosphate are combined to form sucrose. So sucrose 6 phosphate is formed and its reaction is catalyzed by sucrose phosphate synthase. And sucrose 6 phosphate is dephosphorylated. Dephosphorylated means removal of phosphorus or attachment with other. So dephosphorylated to yield sucrose. As you know this, sucrose is a non-reducing sugar and it is made up of glucose plus fructose and it comes under the disaccharides. So however, there are certain studies is also going on, and they have studied these FRKs are indispensable for the carbon metabolism, varying in sense, such as vascular tissues, or you will find in the seeds. Okay, so FRKs are indispensable for carbon metabolism in skin tissues such as vascular and seeds. FRK, FRKs. What is FRKs? See here, you will not find here. Skin tissues such as vascular and tissues. फ्रुक्टोज वहां तक आपका ठीक है. काइनेसिस, काइनेसिस, एटीपी इज इन्वॉल्व. Fructose, FR means fructose. Okay, that I will teach you how it is formed. Okay, आएगा आगे. So sucrose, hexophosphate figure you can see ADP glucose, and this ADP glucose will convert into amylose, and similarly amylopectin, and finally you will find starch. Not like this. There are a number of reaction is happening. Whatever the dotted one, that is the reaction. Okay, that much quantity of reaction is happening. Simply starch to hexophosphate it is not possible. It requires a number of enzyme involved in that. So I am giving you a certain example of ADP glucose. Donor is formed the following reaction: glucose 1-phosphate. Kinase, wherever you will find enzyme, ATP means kinase is there. So glucose 1-phosphate plus ATP, that will convert into a ADP, glucose plus PI. Where? In plastic. In plastic. That is a contrast to cytosol. Now pyrophosphatase, which make the reaction irreversible. Now I am showing you overall reactions. This one is lower one, not like this. Shift plus. Okay. Similarly, here also, when you write this, okay. So glucose 1-phosphate, ADP glucose plus inorganic phosphate, that in cytosols. Now the starch, n number of starch with the glucose 1-phosphate is attaching. Plus ATP is also there. So you will find a storage form of starch, and they have a ADP plus 2P. Taking into account hydrolysis by inorganic phosphatase, of inorganic phosphate produced during ADP glucose synthesis here, right? In the overall reaction, glucose 1-phosphate is, you can write like this, and free energy is changes. Nearly, you can say that minus 50 kg or kilojoules per mole. Okay. As we have studied previously, and I have already told you what is RFOs. RFOs, range forest officer not. Here you have to remember raffinose families, or you can say raffinose family oligosaccharide. So raffinose family of oligosaccharide that accumulate differentially in the different plant parts. Plant shows the variability in the types of RFOs. Raffinose family of oligosaccharide. In contrast to raffinose family, which is universal in the occurrence. STA means stachys and other high degree of polymerization. Now, what are the RFOs? Read.
RFOs such as Bulbacosh, DP5 and Azugoon, DP6 accumulate in the vacuole of only certain plant species.
Very good. That is very important sentence for educations, for questions in the examination can be asked. So you have to remember these. What is RFO? How many types of RFO and which RFOs are found in the vacuoles? Okay. So as you learn, which is the universal one you will find in every plant species. As I said, Stachys. Stachys, Ravenous family part है or it will find in many high degree of polymerization and universally it is occur. But there are two other. One is verbesco and another is ajugos that acclimate in the vacuoles of certain plant species. So raffinose family of oligosaccharide, as I said, there are certain functions or have a wide range of predicted functions. So they are synthesized and stored in the seeds and protecting the embryos. One function is that. What is the functions of RFOs? So they are synthesized only for storage in the seed and for protecting the embryo from the maturation, associated desiccation. Participate in several functions such as transport and storage of carbon, signal transduction, membrane trafficking, and mRNA export. Now, in the examination, if I am asking what is the functions of RFOs in the plant, what is the functions of RFOs in the carbon metabolism, then you have to write it is only synthesized for the various predicted functions according to the species and generally it is protecting the embryo from the maturation associated desiccation. It is also participating in the several functions such as it work as a or it involved in the transport. It is also formed as a storage form of carbons. It also work as a signal transduction. It also involved in the membrane trafficking and it is also work as a export or mRNA export. So that can also be asked in the examination. RFOs are currently emerging crucial molecules, very crucial molecules, where in generally when you are studying with any stress plants, droughts plants, or any research with the stress, you have to study the RFOs. Raffinose family of oligosaccharide currently a crucial role or crucial molecules that during the stress response in the plant. Why? That can also be asked in the examination. Give reason. Because of that, as I said in the function, that it is involved in the membrane stabilizing. Perhaps it also involved as antioxidant and there are a certain predicted signal transductions or signaling functions. If stress is imposed, certain RFOs is formed. Okay. So their role as a reserve carbohydrates, their role as a membrane stabilizer, their role as a osmoregulators, their roles as a stress tolerant mediators. Remember these words. Remember these words for year. If you want to become a scientist in the plant biochemistry, you must read these functions as a stress tolerant mediators because in climate change condition you will find many biochemists will study on these aspects stress tolerant mediators. Now Recently, certain scientists who also proposed smaller representative of these sugars that may contribute to cellular redox potential or redox balances, or it can also act as a phloem signaling compound under stress condition. Remember why I am telling? Because it act as a phloem mobile signaling compound under stress. Now, raffinose synthesis, how it is synthesized in the higher plant, where it is found, which crop is having the raffinose family oligosaccharide, and where it is reported, in which condition it is reported. That already been taught to you. But not only stress condition, any stress condition, it is related with the tolerance to heat, drought, cold injury, disease incidence, or disease resistance. That has also a role that I will teach you in this para. RFO synthesis or raffinose family of oligosaccharide in the plants, mostly D-glucose containing oligosaccharides. How many? D
Galactose.
D
Galactose.
galactose, not glucose. If you speak the D glucose means oligosaccharide, remember maltose plus glucose. Okay, n number of glucose that will comes in the starch. Okay, here is galactose. Galactose containing oligosaccharide in the higher plant. So oligosaccharide are well documented and you will find in many literatures of cereals also, pulses, and देखो cereals and pulses. Another you will find in the fruits and vegetables. So in plants, as I said previously, that is inputted or in part have been reported in the tolerance to heat, tolerance to drought, or cold salinity condition, disease resistant, that regulating also a certain physiological activity in the plant. That is, as I said previously, that is in seed germination, or you can say where it is there, what is happening? सूते गया है.
Pouch कुछ ऊपर में होगा. Pouch.
Now, where I was in the plant, where the physiologically you will find a certain activity because of RFOs. As I said previously, it is protecting embryos. It means during seed germination, it has a role. Okay, see here, regulating seed germination, vigor, and longevity. Longevity means longer storage. You will also found that. So in humans, because of it containing galactose, as you know that galactose is generally found in where glucose plus galactose, glucose plus galactose is equal to lactose. It is found in where? In milk. But I am asking here, in humans, RFOs have a beneficial effect in large intestine. Where? What is the role of RFOs in human?
Beneficial effect in large intestine.
So have some prebiotic potential by promoting the growth of beneficial bacteria and reducing pathogens and putrefactive bacteria present in the colon. It means if you see as a biochemist, if you tell any persons RFO is a nutraceutical food. Why? Because it contains galactose and it has a beneficial effect in the larger intestine, means removal of any stool. So it will reduce the colon cancer. It will reduce the appendix. Okay. In addition to the prebiotic potential, RFOs have many biological functions in humans and also in the animals, such as anti-allergic, anti-obesity, anti-diabetic, prevention of non-alcoholic fatty livers and their disease, and it can also work as a cryoprotection. So this wide ranging of application of RFOs that is also useful in the food preparation, in the paint preparation. You will also find in the cosmetic. It can also work as a health promoter. So many scientists are also working in the pharmaceutical product, and certain scientists are also working in the plants, trees, products. So you can add the RFOs or your treatment of RFOs in the various plant species. So RFOs has subsequently a higher molecular weight. So which are the RFOs? Tell me in the bracket. Read.
Stekios, Verbescos, as you go.
Uh, Azugos. Okay. Ajugos are synthesized from where? From sucrose by a subsequent addition of activated galactose. Okay, that might donate the galactinol. Galactinol. RFOs protect the embryo. As I said, same sentence is here, but writing is different, wording is different, understanding is different. How to write that I will teach you. RFOs protect the embryo from maturation, desiccation, and predominant transport carbohydrate in some plant family. It acts as a signal molecules following pathogens, attack and bonding and accumulating in the vegetative tissue in response to range of abiotic stresses. So this is the, as I said, how raffinose or barbascose or agulose, huh, that galactonol, this galactinol dependent, that is myoinositol UDP galactose, that will convert into a galactinol. So raffinose synthase is the galactinol, sucrose is the main ingredient. As I said, generally galactose is addition with the sucrose that will convert into a raffinose. Galactinol further it is myoinositol is formed with the stachyose synthesis, and stachyose is formed. Similarly, stachyose, after stachyose galactinol, another addition galactinol, you will find verbascose. So galactinol independent and galactinol dependent two pathways are operating. Okay. So generally galactran, galactosyl or transferase is the involved in the raffinose to stachyose and stachyose to verbascose. So you have to remember RSV. What? RSV. An addition of raffinose to sucrose. Nothing else. Same is here. How it is formed. So raffinose family is D-galactosyl of sucrose, a disaccharide. They occur in the form of raffinose, as I said, stachyose, verbascose, ajugose, belonging to trisaccharide. This tetrasaccharide, pentasaccharide, hexasaccharide, this respectively you can say here. Which are the trisaccharides? Okay. So similarly, from the structural perspective, they are considered alpha galactosol derivatives of the sucrose and galactose, glucose, and fructose. Consequently, raffinose contain how many sugar? A cola. Raffinose is equal to
Three sugar.
which are that? Galactose plus glucose plus fructose. So you can say glucose and fructose is equal to what? Sucrose. And the sucrose plus galactose is equal to what?
Sucrose, galactose,
Sucrose plus galactose.
ના એવું કઈ આવતું જ નથી સર
galactose,
Glucose.
glucose plus
fructose का sucrose.
yeah, sucrose plus galactose is equal to
Raffinose था क्या?
raffinose,
हाँ.
right? So therefore, you are not here. I have
Okay.
already told you that here sucrose to raffinose, sucrose to
Sir,
raffinose.
okay, આવતું નથી
Two
પાછું.
times. Yes, therefore, here the raffinose to sucrose, raffinose to sucrose, RSV. Okay. Now I am here. Raffinose contains galactose, glucose, and fructose. Now stachyose. Stachyose hold two galactose. Stachyose hold two galactose unit. One D-glucose and one beta T fructose. Same thing is glucose and fructose is also there. But here you will find two galactose. One galactose means what? Raffinose. Stachyose means two galactose. Now raffinose synthase transfers the galactose moieties from galactinol to sucrose and produce raffinose. While the stereosynthase RSV, remember, stereosynthase utilize galactose to synthesize tetrasaccharide. Both reactions are reversible. However, this enzyme responsible for biosynthesis of verbascose. Yes, has not been recognized yet today. So stereosynthase is the most probable responsible enzyme due to its multifunctional characters. And verbascosynthase, two enzymes, stereosynthase and verbascosynthase. Verbascosynthase is a chief enzyme that catalyzes the synthesis of verbascose by addition of galactosyl residues from galactose to stereose. Nevertheless, synthesis of pentasaccharide verbascose are homologous to galactose independent, and they may include that is alpha galactosalide derivatives and cytosolic myoinositol, D-pentolol, D-chiroinositol, or nonitol. Okay, do nonitol, but with the small amount. Okay, so this is all about the raffinose family and its help in the various plant parts and how it is synthesized. Now I am giving you a RFO or raffinose family oligosaccharide in the plant health. It already been taught to you. I am giving you a certain studies or researcher who have interested in that and where it's what is the role, crucial role in the plant physiology, what is the role in the cellular function or cellular transductions. So as I said previously, it is involved in the membrane trafficking, mRNA export transport. It works as a carbon storage, desiccator tolerance, seed storability. It is also involved in the biotic and aquatic stress tolerance, and it is involved in the phosphate assimilate translocation. This is all about the summary of role of raffinose in the plant. Now, future prospect related to raffinose. As I said, in for we people, for human being, it can work as a functional food. It is multifunctional benefits, as I said previously, that it is yet to be realized in the humans, yet to be realized or multifunctional for the well-being of the animals and humans. RFO have a positive effect, as I said previously, in the major intestine or bigger intestine. It reduces the where it's a positive effect in the gut microflora, larger intestine, and colon health. It has a therapeutic use. It can work as a therapeutic agent, reduce inflammation, reduce diabetes, reduce allergic, anti-allergic, certain anti words already been taught to you. So this is all about the RFO and its roles as a functional food. Sometimes it is also asked in the examination RFO as a functional food. You can also write a research paper RFOs as a functional food. Hmm? Right? You both